All right. Hey, everyone, welcome to the third installment of the NIDA Summer Seminar Series. This seminar is focused on the behavioral pharmacology and the neurobiology of substance use disorders. And in addition to my lecture covering behavioral pharmacology, we have a guest today, Dr. Sunila Nair, who's going to be covering neurobiology of substance use disorders. So we're going to start with my section on behavioral pharmacology. So what is behavioral pharmacology? So behavioral pharmacology is the study of how drugs affect the behavior of organisms, so both humans and non-human animals. And it's a relatively new science, dates till probably about the 1950s, and it's based on the integration of the theoretical and scientific principles of basic experimental pharmacology and radical behaviorism or behavior analysis, also known as sort of Skinnerian behaviorism. So the school of thought there is that it's a data-based science where the focus is on behavior and experimental manipulation and control of behavior in order to understand behavior-environment relations as mechanistic processes. It's distinguished from neuropharmacology and neuropsychopharmacology, which also emphasizes the mechanistic relations between behavior and its biological or physiological substrates. So traditionally, behavioral pharmacology largely treats what's going on inside the central nervous system as sort of an irrelevant black box. That's not so much true anymore, but the reason why historically it's done so is at the time in the 50s, we just didn't have the scientific toolkit to be able to really study the neurobiological substrates or mechanisms that drive operant behavior. So the idea was that simply focusing on measuring and manipulating environmental variables that affect behavior and then behavioral output was a clearer, more parsimonious way to study the effects of drugs on behavior. So behavioral pharmacology really developed in parallel with neuroanatomy, neurophysiology, and also the neurobiology of learning and memory. And so the synthesis in these advances really spawned the discipline of modern neuropsychopharmacology. So when I'm talking about different features from those fields that it brings together some basic tenets of experimental pharmacology. So a lot of human behavioral or just behavioral pharmacology studies in general, in both humans and non-human animals, one sort of paramount feature is either experimental administration of drugs under controlled conditions or also examining self-administration of drugs, usually drugs of abuse under controlled conditions. And sort of two major subfields of assessment from pharmacology that are relevant here are pharmacodynamics. So that is essentially what the body does to the drug. So the way the drug is absorbed, the rate at which it enters usually the bloodstream and then crosses the blood-brain barrier into the central nervous system, which is the effect compartment of psychoactive drugs. How it is distributed throughout the body, how is metabolized, so how levels start to decrease after reaching their peak biological concentrations, which are usually closer to their peak behavioral effect concentrations, and then how it's excreted afterwards. It's also the basic principles of pharmacodynamics. So studying the way drugs that are experimentally administered to organisms, studying their effects on physiology, so changes in heart rate, blood pressure, respiratory rate, etc. Changes in behavior, which can be anything from simple analogs, grosser analogs of behavior, so the amount an animal moves, the amount it sleeps, or human, to more specific behaviors that have implications in understanding the etiology of drug abuse and the effects of drug abuse on public health or individual health. So things like the way experimentally administered drugs may affect decision-making about choices related to spending money or having risky sexual intercourse, etc. So those are things that are also examined. It will also, pharmacodynamic effects, we look at the effects on intraceptive or essentially self-report internal states. So those would be subjective drug effects. So things like feeling high or feeling anxious, things that are sort of intraceptive or internal reasons why humans report using drugs. One other critical component here is the examination of dose-response relations. So in general, with almost all psychoactive drugs, you get something called a dose-effect curve, where at levels or doses that produce levels below a threshold, you won't see any effects. Then as doses increase, you will see an increasing range of effect with increasing dose until you generally hit a maximum effect range for that given described effect. So you can have a different dose response relation for behavior A versus behavior B. Like for example, you may have a dose response relation for positive effects or feeling high that that parallels but does not completely line up with a dose response relation for being sedated or feeling anxious. And oftentimes what you have with most drugs of abuse is the dose response effect curve for positive subjective drug effects, or the beneficial ones, is usually shifted to the left relative to the adversive ones. So generally you will hit high levels or maximum levels of desirable drug effects at lower drug concentrations or doses than you will for the adversive or negative effects, which does a lot. It sort of demonstrates why human beings, when they drink or use other drugs, will generally self-administer doses within a certain range and often avoid higher doses. So behavioral pharmacology also brings in some basic principles of learning and behavior, both classical and operant conditioning. So classical conditioning, also known as Pavlovian learning, refers to the processes involved in non-voluntary or passive learning. So the classic arrangement would be, the Pavlov dogs experiment, where a dog will naturally salivate to a potent biologically relevant stimulus, such as the sight of food. Now, after repeat pairings of, the sight of food. Okay, without any conditioning, a dog will not salivate to the sound of a bell naturally, right? But through repeated pairings of the unconditioned biologically potent stimulus with the neutral stimulus of bell, over time, the animal will learn to associate that previously neutral stimulus with the unconditioned stimulus, the food, and the bell will start to elicit the salivatory response that the dog would naturally get from food. So the sort of biological purpose of this learning mechanism is to allow organisms to learn about their environment and learn how other stimuli predict biologically potent stimuli in order to function better in the natural world, right? So to learn about predictive relations between, the occurrence of one stimuli predicting another, right? The sound of hoofbeats predicting a stampede of wildebeest that you might want to run away from, right? So in addition to classical Pavlovian aspects in drug use, another and perhaps sort of more germane foundation is understanding the principles of operant conditioning or instrumental learning. So this refers to how voluntary behaviors are controlled or modified by contingent consequences. So consequences that happen as a result of their occurrence. And in general, these consequences can sort of be divided into two categories that are in fact defined by their effects on the behavior. So reinforcers are consequences that increase the future occurrence of the behavior, and punishers are consequences that decrease the future occurrence of the behavior. So reinforcers can be both positive or negative. So it can be the delivery of a pleasant consequence or the removal of an unpleasant consequence. Likewise, with punishment, punishers can be the delivery of an unpleasant consequence for the behavior or the removal of a pleasant stimuli or withholding a pleasant consequence. So one of the sort of paramount experimental paradigms in behavioral pharmacology is the study of drug self-administration. So drug self-administration studies, again, are viewing drug use as a voluntary behavior governed by its reinforcing properties or aversive properties, but in general, just its consequences. So in these arrangements, generally, so this can be done with animals or humans, under controlled conditions, the organism will learn that they're able to perform a voluntary operant response, pressing a lever, a button, etc., or requesting drug from a research assistant in a human paradigm, and that they will receive a dose of drug contingent on performing that response. So the figure on the top right is an example of a Skinner box or operant chamber, simplified example where this rodent has been trained to know that if they press the lever on the right, it will deliver an infusion of the drug under a given schedule reinforcement. So the way these data are generally recorded, you'll see in the bottom figure, it's an example of a cumulative record. So during an experimental session or across experimental sessions, it's essentially a ticker tape. And every time you get a response, it bumps up 1 click. So what the cumulative record curve right there does is it allows you to record not only the cumulative number of lever presses for the drug during a given experimental session, but you can also see during what parts of the session the lever pressing is more intense or the responding is more intense. You can tell by a steeper slope of the curve, and then where it's largely leveling off as the curve gets more gradual. So with drugs, along with a lot of other reinforcers, there are substantial differences or effects of schedule of reinforcement on the rate of responding. So the most simple schedule would be a continuous schedule of reinforcement. That's where an organism has delivered the reinforcer in germane to this example drug as a function of every time they perform the operant behavior. So this is a real good way to train an organism in self-administration, and it reliably establishes self-administration behavior of a desirable reward, or it will reliably establish avoidance behavior of a fairly robust punisher, meaning that training an animal to lever press to avoid a shock or other unpleasant consequences. Both of these are reliably established with a fixed interval, fixed ratio schedule or continuous schedule. But you generally don't get very high rates of pressing because animals like humans often satiate with a given reinforcer. So the idea is, if you were hungry and you had to press a lever once, every time you pressed it, you got a hamburger. Well, once you're up around 4 or 5 hamburgers, you're not going to be hungry anymore, and your rate of responding is going to decrease. Not necessarily because hamburgers aren't reinforcing for you per se, but because you've had 5 hamburgers and you're not hungry anymore. So one way to sort of study behavior in a way that makes it less sensitive to satiation effects is to look at different schedules of reinforcement. So things like variable interval, meaning that the response is reinforced after a variable amount of time, or a variable ratio, meaning that's reinforced after a variable number of responses. So variable ratio schedules where the reinforcer has a variable magnitude, meaning you're not quite sure how large it's going to be, end up producing some of the highest rates of voluntary behavior per the amount of reinforcer delivered overall. So one example there would be human beings that have a tendency to gamble or play the lottery, right? So the payout on scratch tickets or lottery tickets is usually 5 to 1 or 7 to 1. But you don't know when you're going to win, and you're always winning when you do win different amounts, and occasionally the amount's going to be fairly large relative to the response cost or that dollar you paid to buy the ticket. And because of that, you get a large proportion of individuals to allocate a substantial amount of behavior to that activity. And it's largely a function of the ratio and the variability in magnitude. For example, if you always just won $1 back every 5th time, right, which is the even payout rate, you would get very few people who would play the lottery, even though that's how it breaks down, ultimately over the grand scheme of things. So that's an example of how the schedule of reinforcement in itself can put a dramatic amount of control over behavior. And this also applies to human drug use too, because if you think about it, if you are often buying drugs illicitly on the street, you're getting drugs of sometimes unknown quantity for your money or unknown potency. So, if you're buying a $40 bag of cocaine on the street, maybe sometimes it's going to be particularly pure. Sometimes you might get a particularly fat bag. And under those conditions, that variability tends to help maintain higher rates of behavior in many cases for some individuals. The other sort of schedules of reinforcement used in addiction research are progressive ratio schedules, or something called a breakpoint assessment. So that's a schedule of reinforcement where the amount of work or number of responses for a given drug or dose of the drug is increased over time. So it will generally be like, okay, so for a given dose of the drug, let's see if the animal will press the lever 2 times. Okay, if they press it 2 times, let's see if they'll press it 4 times for the next dose. Okay, then 8. Okay, then 16. And that will increase until you get to the highest point where the animal will essentially stop pressing. And what they're essentially doing there is showing you equivalence or functional equivalence of how much work that dose of the drug is worth. So you may have a condition where it's like, okay, so this animal will press a lever 256 times for this one dose of IV cocaine, but it won't press it 516 times or 512 times. So essentially what they're saying is that the amount of work done, so 216 lever presses is functionally equivalent in value to the rewarding or reinforcing properties of that one dose of drugs. You also have some experiments that use second order or chain schedules. So without getting too much into these are more complex arrangements that tend to be difficult to establish experimentally, but what they do is they do a really good job of modeling real world human drug use behavior, because through Pavlovian or classical conditioning and operant paradigms, you can essentially shape up an experimental model of human drug seeking behavior where it's often, the initial behavior in the repertoire isn't using the drug. It's often, tracking down a phone number to call someone, to take the subway somewhere, to et cetera, et cetera. So these are experimental arrangements where, with the drug being the primary reinforcer, they can backchain and actually train laboratory animals to lever press to be able to see a light that has previously signaled access to the drug, or perhaps even another step in the chain, a light that signals the availability to press a lever for a tone, that tone previously being associated with access to the drug. So these are really procedures that show the continuity between animal behavior and human drug use behavior and further validate these as experimental models for studying addiction. There's also extinction and reinstatement training, which basically extinction of responding would mean that the animal's previously trained to lever press or human button push for a drug, and then the response stops delivering the reinforcer. So essentially looking at how long it takes for that animal to stop responding when the reinforcement's removed. And then reinstatement would example the resurgence of responding, how quickly it happens once the drug becomes available again. So this is one way to model relapse. Another common procedure done is dose response testing. So this essentially looks at pressing on different schedules. This is often a continuous or fixed ratio schedule for different doses of a drug alone or in combination. And what you see here, you see that on the bottom right for both, example figure for both cocaine, heroin, and their combination. And what you see here is a classic example of what you call an inverted U dose response curve. And what that shows is that as doses increase, generally as the drug gets more reinforcing, the number of infusions chosen decreases. Then once you get over a certain dose, it tends to decrease. That's not necessarily because one of those single doses is less reinforcing. One of those single doses may in fact start to become aversive. But it's generally just that the repeated or total amount of dose delivered across multiple boluses starts to get non-reinforcing. So at very high levels, individuals will reduce the amount of work they're doing per drug infusion because they're essentially satiated or it becomes aversive. So all these are procedures that model discrete aspects of real-world human drug use behavior. In addition to looking at simple schedules of reinforcement, we often look at choice procedures. So instead of assessing the magnitude or intensity of responding for a single reinforcer, choice procedures examine allocation of behavior for responding across both drug and non-drug alternative reinforcers. So this is perhaps more analogous to real-world behavior in human drug users than responding for just drugs alone under different schedules, right? So a lot of times in these drug-only response conditions, the animal can, essentially lever press for a drug or do nothing else or engage in grooming behaviors, et cetera. When, humans in general, we're making choices every day between a lot of different things. We're choosing to, eat lunch instead of go for a walk, or go to a movie instead of stay home and save money, et cetera. So these choice procedures do a much better job of capturing real world behavior. And they're consistent with something called matching law, which is a simple model developed by Richard Herrmann that just demonstrates essentially that the proportion of time dedicated to performing a specific behavior is in general directly proportionate to the total amount of reinforcement in the organism's environment offered by that behavior. And what this goes to explain is the reason why drug self-administration in substance use disorders as they progress to addiction tends to take up a particularly large amount of a person's time, is that as you spend more time responding for drugs, your other natural reinforcers, your non-drug alternatives, tend to drop off. So as you're responding for drug increases, you may be losing other friendships with non-drug users. You may be, through changes in your health, be losing the ability to play certain sports or engage in other activities. So it sort of creates that slippery slope where responding drifts more and more strongly towards drug-associated responding because the other reinforcing alternatives in your life tend to drop off. So this is something we've shown that's consistent across species, and it's sensitive to medication effects as well. Just looking at how I'm doing that time. So what we're looking at here is on the bottom are choices in monkeys between different amounts of food and different doses of cocaine. So 1 pellet of food versus cocaine in this figure A, 3 pellets versus cocaine in figure B, and 10 pellets versus cocaine in figure C. So what we're looking at here is across trials, the number of choices for either cocaine, food pellets, or admissions. So remember, the organism can do nothing. So what we're seeing across these 3 figures, just is something interesting, is that as the dose of cocaine increases at a given food magnitude, so for 1 pellet, choices for cocaine over food pellets or admissions tend to increase. Now, at an equivalent dose of cocaine, so if we look at the second group of bars, the 0.043 across the 3 figures, A, B, and C, we see that as the magnitude of the alternative food reinforcer increases at a given dose of cocaine, responding for food increases. And then we see as both increase proportionately, we see very few emissions at high doses. And in this case, it seems like this higher dose of cocaine is a more potent reward than even the 10 pellets of food. So what we're seeing is that the choice behavior is sensitive to manipulations in magnitude across both the drug reinforcer and alternative reinforcer. And this is also very, very analogous to real world behavior. So again, these studies have shown that we see, similar types of results, whether we have an animal, lever pressing. These data are displayed a little differently. An animal lever pressing for cocaine responses versus food, or an operant arraignment where a human is choosing between a dose of intranasal cocaine and different amounts of money as an alternative. So this would be an experimental study where humans come in the laboratory and they're allowed to sample a drug, and then they're given choices between using small amount of that drug right in the laboratory or receiving an alternative reinforcer, in this case, various amounts of money ranging from zero, so no money, 50 cents, $1, $2. And what we see here is that as the amount of alternative reinforcer increases in that laboratory session, the percent of choices that are allocated to cocaine decrease. So behavioral pharmacology, one of the most sort of, I think, useful applications of these methods has been as a medications development tool. So as I said earlier, there are studies that have demonstrated that drug self-administration and choice is sensitive to medication effects. And a lot of this is back translation. So some of this is research that's been done with medications that were developed through sort of trial and error testing that we've shown prior to behavioral pharmacology, that we've shown work in the clinic at reducing drug use. We've shown that these models show the same effects. So medications that work for opioid use disorder, for example, methadone, in the real world, we know that they reduce drug use in patients in treatment. Also reduce responding for drug in this type of choice procedure arrangement. So this is an example from this is a human study looking at responding for placebo and placebo on where it says 0 milligrams of heroin, 10 and 20 milligrams of heroin over various amounts of alternative money. So again, this is a series of choices between, would you like this dose of drug right now or this money? Consequence, they're delivered at 3 different doses of methadone. And what we see here is that a reliable effect, as I said earlier, of as you increase the magnitude of the drug reinforcer at a given, for a given magnitude of alternative reinforcement, drug choices increase. As you increase the value of the alternative reinforcer at a given dose of drug choices decrease. And then as you put on this medication that's known to reduce drug use in the real world, it also has the same effects under these laboratory conditions. So what this does is this shows us that we have a tool to rapidly screen potential medications in the human lab versus waiting to do a 5-year clinical trial to test every new potential medication to see if it works. These are studies that can be done with a dozen or so participants in the lab. So I was gonna go through an example of one of those studies that I worked on, but I think I'm gonna skip that for the sake of time because we're getting close to 3:30. So I'm gonna cut through all this. So another alternative way of looking at the reinforcing effects of drugs are conditioned place preference paradigms. So this is an alternative measure that doesn't involve direct responding or drug self-administration. This essentially is a measure of the reinforcing effects of drugs that relies on both Pavlovian or classical conditioned associations and the operant response of simply where the animal spends its time. So prior to the conditioning procedure, the same animal will be in a chamber, as we can see up the top, divided into 3 sections. So there's a middle and there's a left and a right. So the left and the right, so this animal has no experience in there with drugs or anything. It hasn't been paired with anything yet. But they are distinguished by salient environmental cues. So maybe the chambers are a different color or have different color lights illuminating the top of them or different odor. So then in one side of the chamber, the animal will be handled and injected with drug, or drug will be delivered by the experimenter. And then on the other one, perhaps they will get injections of placebo or sometimes nothing at all, but generally a placebo control. And then after numerous pairings of drug injection occurring on one side or one place in the chamber and placebo occurring on the other, the test session will allow the animal to sort of spend time in the chamber and we'll see where they hang out. And the idea is that if the drug is functioning as a reinforcer, so if they associate that delivery with positive effects, they're going to go and spend more time in that side of the chamber because that side of the chamber is associated with delivery of the drug. Likewise, if that drug's aversive or unpleasant, they're probably going to avoid that side of the chamber or spend time in the middle or in the neutral side. So this is a lot easier to establish than drug self-administration. It takes a lot less time in terms of running these experiments because it's oftentimes really hard to train animals to reliably self-administer drugs. But the one downside is it may be sort of less informative than drug self-administration procedures in terms of its predictive validity of medication effects or other aspects of real-world behavior. So just some summary points. So behavioral pharmacology is a powerful experimental science that's contributed greatly to our understanding of substance use disorders and the development of treatments. And many of these advances were largely accomplished, largely independent of studying the neurobiological mechanisms that underlie these behaviors. So the focus is largely on manipulation of environmental contingencies and the measurement of behavior. So integration of behavioral pharmacology and modern neuroscience and neuropsychopharmacology is really sort of a newer science emerging in the 80s and early 90s. And that's what's really leading to further progress in the area. So being able to understand the underlying neurobiological substrates that drive these behaviors is allowing us to make deeper and more meaningful advances in understanding drug use and its treatment. And that will be my segue into our second presentation.
Thank you, Evan. I'm going to share my screen. Can you see this?
Yep.
Okay, great. So as Evan said, I'm going to talk to you today about the neurobiology of substance use disorders. And just to go over this briefly, I know you have talked about it before, but we know that addiction is a really debilitating and costly neuropsychiatric disease that is characterized by uncontrollable drug craving, compulsive drug taking and drug seeking behaviors, and very high propensity to relapse. Approximately 40 to 60% of individuals will relapse at some point. But despite this, only about 10% of patients are in treatment, and this is largely due to the stigma that is associated with the disease. And you've also talked about how substance use disorders occur as a continuum. So an individual may have an opportunity to use a substance, and this might develop into the experimental use of a substance. And in some individuals, it might progress to established use. and a full-blown substance use disorder. And eventually an individual may seek treatment, but like we just talked about, they may relapse to substance use and then go into remission, and the cycle can keep going over and over again. So what we're going to talk about today is what are the changes in the brain that happen when an individual is exposed to a substance acutely, and then how do we study brain changes as the disease progresses from acute use to habitual use and eventually to relapse to substance use disorders. So what is it? What is the neurobiology? What is it that makes substances addictive? So to understand this, we're going to start by talking about the pharmacology of the neurotransmitter dopamine that I'm sure you all are familiar with. So if you look at the panel to your left, panel A, that's a neuron under basal conditions, it's a logical condition, so without any drug, the dopamine neuron. And the presynaptic terminal of the dopamine neuron has the neurotrans has dopamine in synaptic vesicles. And when an action potential strikes the presynaptic neuron, you see that the vesicle gets fused to the membrane of the presynaptic terminal and releases dopamine into the synaptic cleft, where the neurotransmitter can bind to receptors that are on the postsynaptic terminal and stimulate second messenger cascades. And then the dopamine that is in the synapse also gets taken up back into the presynaptic terminal by the transporter, the reuptake transporter that you see in yellow in the membrane of the presynaptic terminal. So that's under basal conditions. So what happens when there is cocaine on board? And that's what you're looking at in the middle panel B. So when there's cocaine on board, it binds to the reuptake transporter. So when that happens, the dopamine that is in the synapse, it cannot go back into the presynaptic terminal and get packaged into the vesicles again. So what this leads to is this massive increase in dopamine in the synapse. Similarly, with amphetamine, the mechanism is slightly different. Like amphetamine, it actually it has a higher affinity for the dopamine transporter compared to dopamine itself, and it gets taken back up into the presynaptic terminal instead of dopamine. But the end result is basically there is an increase in dopamine in the synapse. So we'll take a minute and talk about what dopamine pathways in the brain are. And they're basically classified into three distinct pathways. And what you're seeing in blue is the mesocortical pathway. And this consists of neurons with cell bodies in the midbrain, and it sends axons to different parts of the cortex. The mesocortical system is basically responsible for cognitive function, memory, attention, emotional behavior, and learning. The next up is the nigrostriatal dopamine system, which you're seeing in green, and it has cell bodies in the substantia nigra, and it sends axons to different parts of the striatum and is responsible for movement and the integration of sensory stimuli. And then finally is the mesolimbic dopamine system that you're seeing in pink, with cell bodies in the midbrain, and it sends axons to different parts of the limbic system. And this is the pathway that is responsible for pleasure and reward-seeking behaviors, and also addiction. So we talked about how addictive drugs increase dopamine. I want to take a step back to point out that natural rewards also produce the same response. They produce an increase in dopamine levels in the synapse. And these are experiments, data from experiments that were done using microdialysis, which is very old. It has been used for decades. The technique has been used for decades, where you put in a dialysis probe. It's a probe that has a semi-permeable membrane. So the membrane goes into the brain region that you're interested in, and then it is perfused by a fluid like artificial an artificial cerebrospinal fluid. And so dialysis occurs across the semi-permeable membrane, and you collect the dialysate and then analyze it for neurotransmitters with high performance liquid chromatography or mass spec and other techniques. So basically, you're measuring the neurotransmitter content in a specific brain region in a specific period of time. And so what you're looking at on the left is the dopamine that is released in the nucleus accumbens, which is part of the limbic system in response to food. And there is a significant increase in dopamine when an animal starts eating. Similarly, looking at the data on the right, during a sexual encounter, there is also a significant increase in dopamine levels, suggesting that natural rewards also increase dopamine. But what I want you to appreciate is basically the magnitude of the difference in this dopamine release. So in this figure on the left is, like we just talked about, there's about a twofold increase in dopamine during a sexual encounter. And on the right, you see that there is about a tenfold increase in response to amphetamine. So there is like orders of magnitude difference when an animal is exposed to an addictive substance as opposed to a natural reward that can also increase dopamine. And that is what makes drugs so addictive. So then I'm going to move on and talk about cells and circuits in the brain and how we can study this with respect to motivated behavior. And I just wanted to appreciate how complex and interconnected this network is. And this is a picture by Ramon Cajal. So it is a very old, century-old picture, but it has been known for a very long time how complex this brain is. And a quote from him says that the brain is a world consisting of a number of unexplored continents and great stretches of unknown territory. And this is true. This is true even today. And just to give you a perspective, the human brain consists of 86 billion neurons, and just 1 cubic millimeter of tissue contains 50,000 neurons. Each of these neurons can connect to 10,000 other neurons and result in a massive number of synapses, which are communications between neurons. So that is how complex and interconnected the brain is. So that is probably why it has been so difficult to develop treatments for neuropsychiatric disorders, because of the complexity that we're dealing with here. So with respect to substance use disorders, what are we trying to understand? We're trying to understand key cell populations that may drive these motivated behaviors. What are the inputs and outputs to these key cell populations? And what role do these cell populations play in the transition from acute exposure to a drug to habitual behavior and addiction and maintenance of the behavior, and then going through withdrawal and eventually relapse? So we'll talk a little about the tools that are available to modulate activity of cells and circuits, and also some newer tools that are available to visualize cellular activity. But first up, I'll just take a minute to talk about conventional techniques that have been used really for decades to study the role of discrete brain regions in motivated behavior. And you're probably familiar with lesion studies, electrical lesions, and chemical ablation studies, which basically take out an entire brain region, and then you study the effect of that manipulation on, say, like what haven't just talked about, cocaine self-administration, progressive ratio, extension reinstatement, and different measures. And then there are pharmacological tools which have been used for decades and continue to be used with, for instance, site-specific injections of various agonists and antagonists, receptor agonists and antagonists, and to see how modulating a specific receptor, the effect that modulating a specific receptor has on motivated behavior. And then there are electrophysiological techniques and neuroanatomy, which really never gets old. It's really critical to understand neuroanatomy to be able to understand like what the brain, the role of a specific brain region or circuit is in motivated behavior. So I'm going to start off with talking about chemogenetics, which is very commonly used these days. And it is used not only to understand the role of discrete neurons in a discrete brain region or glia specifically, but also the role of projections from one brain region to the other. And it is really based upon a very simple principle that neurons signal via receptors. And we talked about this briefly earlier, that neurotransmitters released from neurons, they bind to receptors on the postsynaptic side, and this results in downstream changes in the activity of various cells and circuits. So the most common type of chemogenetic tools are called DREADs, which stand for designer receptors exclusively activated by designer drugs. And this system was developed by Dr. Brian Roth at the University of North Carolina at Chapel Hill several years ago. And so what these receptors are mutated muscarinic receptors. And what the mutation does is makes the receptor lose affinity for its native ligand. So in this case, the native ligand is acetylcholine. So this receptor no longer binds to acetylcholine. And instead, it binds to a ligand which is inert, and that can be injected by us. So we are able to control the activity of these receptors. So this inert ligand can be clozapine and oxide, clozapine, and there are several other newer ligands available as well. So this is a system that is very commonly used to modulate the activity of cells, and based upon which receptor is expressed, it could be a inhibitory receptor, which will result in the silencing of a brain region, or it could be an excitatory receptor, which will activate a brain region. So based upon what kind of designer receptor you're using, you can modulate the activity of a brain region in a fairly nuanced fashion. And so how do you put this receptor into the brain? There has to be a tool to put this receptor into the brain, and viruses are those tools. And there are several different kinds of viruses that are available. And I'm going to talk about one system in particular that is used very commonly in conjunction with chemogenetics and also optogenetics, which is adeno-associated virus. So what you see, the figure in the right, is a brain region that is infected. So the neurons in the brain region that you see in DREAD, these are neurons that are infected with a virus that express these DREAD receptors. And this is how you inject these viruses. It is a fairly straightforward system that many of you might have in your labs, in the labs that you're working in. So there is an injector, which is connected to a tube, and it's connected to an injection needle, which is attached to a stereotactic apparatus. And you put your experimental animal and inject the viral vector into the brain region of your interest. So there are several advantages to using this adeno-associated viral gene transfer system. It is not popular for which is why it's really popular. The advantages are that you get very high titers from this viral vector. The toxicity levels are relatively low. It is widely available commercially. The expression is relatively long-term. It can last for months. It is relatively easy to package, and it is available in multiple serotypes, like like how we have different ice cream flavors. There are different serotypes, and there are advantages and disadvantages to each serotype. However, the payload that can be packaged in an AV system is relatively small. It is slow to turn on. It typically takes a couple of weeks. It might get integrated into the chromosome, and there may be lab-to-lab variability based upon how you're making and packaging the viral vectors. And the people think that it is super infectious. There are instances when that might not be the case, which I'm going to talk about right now. So like I said, there are different serotypes of the virus, and that depends upon the capsid that is on the viral vector. And what I'm comparing for you today is a type 5 AAV and a type 8 AAV vector. And this is from infecting neurons in a brain region called the lateral habenula, which is responsible for negative reinforcement. And you'll see it's really obvious that the type 8 viral vector has really high infectivity compared to the type 5 viral vector. So that is one thing that you should think about when you're using adeno-associated vectors is the serotype of the virus. So here's one example where we have used dreads packaged with adeno-associated viruses to understand the role of a brain region in motivated behavior. And these are experiments from cocaine self-administration that Evan just talked about. This is on a FR1 fixed ratio 1 schedule, where the animal is making 1 lever press and getting 1 injection of cocaine. And so when we injected dreads into this brain region and injected the inert ligand, clozapine and oxide, what you can see on the right-hand side is that there is a significant increase in lever pressing when the animals get injected with CNO. So basically, when we silence this brain region, there was a significant increase in cocaine self-administration. And this is just one example to illustrate how dreads can be used to modulate motivated behavior and to understand the role of a specific brain region and cells in that region in a behavioral phenotype. So that was just understanding the role of a brain region. And what we like to do, and once we know that, we like to get into the circuit. So every brain region projects to another, which might project back and form a circuit. So how do we study the role of brain circuits in motivated behavior? And systems that are very commonly used these days are intersectional techniques or techniques using dual viral vectors. And so to explain this, again, I'm going to use the lateral habenula as an example. And this brain region, it projects to several other targets. And an important target is the ventral tegmental area, which contains cell bodies of dopamine neurons. And so if you want to study this specific projection from the lateral habenula projecting to the ventral tegmental area, one way to do it would be to inject a inverted viral vector. So this is an inverted dread that you inject into the lateral habenula. And then a second viral vector is injected into the target region. And this is a canine adenovirus that expresses Cre recombinase. And the beauty of canine adenovirus is that it gets transported retrogradely. So when you inject it into the ventral tegmental area, it gets taken up by cells in the ventral tegmental area and it gets transported back into the lateral habenula, where it flips the inverted dread into its proper orientation and it allows for transcription to occur. So when you inject CNO, what you're studying is the effect that you see is the effect of modulating this specific circuit from the lateral habenula to the ventral tegmental area. So I'm going to skip this. Optogenetics is a tool that is used very commonly, but I'm going to skip it in the interest of time. And then in vivo fiber photometry and calcium imaging is a relatively recent technique that is used to visualize cells in real time. So not only are you examining the role of a brain region or a circuit in a behavioral effect, but you're actually visualizing a specific cell and you're able to see what happens to that cell as the animal progresses from drug self-administration, what the cell does during withdrawal, and eventually even during relapse. So this is a really new technique, but a really powerful way to understand the role of specific cells or a specific group of cells in the brain region across different phases of motivated behavior. So I'm going to switch gears a little bit and talk about an understudied area in neurobiology, and that is sex and gender differences in addiction. And so there are several studies that have shown, both preclinical and clinical, showing that escalation of drug use is significantly greater in females. The transition from drug use to addiction is more rapid in females. When women seek treatment, they're consuming significantly greater quantities of drug compared to males, and there are greater reports of craving and relapse in females. And this has been shown in preclinical studies using the models that Evan talked about, self-administration. It has been demonstrated using progressive ratio schedules and extinction models and reinstatement, withdrawal, etc. And I'm going to show you data from a model that is relatively less studied, which is called incubation of craving. And I'm going to quickly explain this. So here, animals are trained to self-administer a drug for about 10 to 12 days. And eventually, they go back to the home cage and they are brought to the self-administration context at different time points after self-administration. And so that's the figure on the left-hand side. So what you're looking at on the y-axis is the number of lever presses, and on the x-axis is gross withdrawal days. And you'll see that there is a time-dependent increase in lever pressing across withdrawal days. And this phenomena is known as the incubation of craving. So craving increases over time, which is a little counterintuitive to what you might think. However, this is not a permanent effect. As you can see from the figure on the right, it peaks at about 30 to 90 days. This is specific for cocaine. And eventually it comes back down to baseline in a few months. And this incubation of craving is also seen in humans. It's not specific to preclinical models. So you can see on the left, this is the craving score on the y-axis, the craving score from individuals who are in an inpatient clinic, which decreases across days. However, there is a significant increase in the days after discharge. And there is a similar effect that you see with cigarette craving on the right. So we were interested in understanding sex differences and incubation of cocaine craving. And there are no, and I'm going to just concentrate on the figure on the right, which is looking at the expression of incubation. And you'll see that there is a significant increase in incubation in males, which is shown in blue. But what was very striking to us is that we saw a significant potentiation in incubation in females that were in the estrus phase of the hormonal cycle. So just like humans, women go through a 28 or 30-day menstrual cycle. In rats, there is a 4-day estrus cycle. And we saw this potentiation of expression only in animals that were in the estrus phase, where estrogen levels are really high and the animals are ovulating. So this is very interesting. And there are several studies which suggest the role of gonadal hormones in sex differences in substance use disorders. But with respect to neurobiology, understanding the cells and circuits or receptors and ion channels that drive this phenomena, it's really something that is not well understood and needs to be studied further. So I'm going to skip this in the interest of time, and I'll leave you with some questions about what the study of neurobiology is telling us. So preclinical data really suggests that the best targets will be highly specific, either subpopulations of neurons or portions of circuit that can help attenuate drug taking and drug seeking behaviors. So how can we use these tools that are developed in the lab? How can this be translated to people? So that's something to think about. And can this lead to the development of targeted drug therapies or even reversal of neuronal changes and a potential cure for substance use disorders? So I'm going to stop there, and maybe we can spend the rest of the time on answering questions.
Evan, there is a question in the chat.
The graduate program recommends study. Okay, so I mean, that's something Albert's gonna go into some of this in his session on NIH Reporter, but I guess some general advice would be, That's a very specialized area for a program to have a programmatic focus. What you might want to do is look at individual investigators, either by their publication record or their faculty profile, or what grants they have funded that are currently doing that type of work. Generally, folks that have a history of doing several studies in succession in that specific area, and probably reach out to them and see if they train graduate students and if there are any that they would be taking. So rather than start the search at the program level, you'd probably want to start the search at the scientist level and see if they're a group that trains grad students, that has a history of training grad students, that may have access to a NIDA training grant like a T32, and has trained students in these areas. So that's the approach that I would recommend, considering that's a pretty specialized area. Neuroscience, yeah, probably. So preclinical med development type stuff, yeah, that's probably more of a neuroscience type program or behavioral neuroscience program. In terms of pharmacy, that may be more, so research in pharmacy programs, a lot of that's more patient access based or delivery. So that would be, if it was something you were interested in methods to increase, interventions that increase access to naloxone in the community and stuff like that, then yeah, maybe more of a pharmacy program. Although there are some pharmacy programs that do basic preclinical work like that, but that strikes me as something of more of a biomedical sciences or neuroscience program. Anybody else? Not everyone at once now. Yeah, so again, if anyone, you all have my email address as well. If anything else comes up or you think of any questions over the next couple of days, please feel free to reach out directly to either one of us. Yeah.